Hello and welcome to Inside Exercise. I'm Emeritus Professor Glenn McConell. Today I'm bringing to you Professor Jørgen Wojtaszewski from the University of Copenhagen. He's an absolute expert on glucose uptake during exercise and also on how exercise increases insulin sensitivity. So we start off talking about what is insulin resistance, what is insulin sensitivity, and then we talk about glucose uptake during the actual exercise, and then how exercise actually increases your insulin sensitivity. So it increases your glucose uptake in response to insulin after exercise. We talked about how exercise increases insulin sensitivity, so the mechanisms involved. We talked about how long the effect of exercise lasts for, the effect of different types of exercise, whether there are sex differences, the effects of aging, and we even talked about whether one day there may be an exercise pill. So a way to try and mimic the effects of exercise to increase your insulin sensitivity. I really think you'll enjoy this one, so stick around. Hi, Jorgen, how are you? Welcome to Inside Exercise.
Good morning, Glenn. Thank you for inviting me.
Yes, great to have you on. So today we're going to talk about exercise and insulin action, yeah? So first of all, what I quite often like to ask people is, did you come through like an exercise background or a science background? Were you like a scientist who developed interest in exercise, or the other way around? How did you get into this?
Yeah, well, for 25 years ago, we had this Copenhagen Muscle Research Center that was placed in Copenhagen, and I grew up in that area, or in that center, and therefore I think I come from a muscle exercise background with very prominent researchers around me that influenced my career greatly. So, but then you could say that a little later during my fellowships, I went to Joslin Diabetes Center because I had this interest in insulin as well, and insulin action, but in particularly the sort of interaction between exercise and insulin. So that was my entrance, and I sort of bridges two fields in a way, the exercise field and the insulin field, for good and for bad. I always felt that I belonged to one at some point, or not. I sort of have a leg in both of these areas, and I have been reaching.
It's complicated, isn't it? Because when you apply for grants, you want to talk about exercise, you want to talk about insulin, then you talk about exercise's effects on insulin.
Yes. But it turned, in sort of the old days, you didn't think about exercise in medicine or in health promotion. Of course you did a little bit, but it was not a big issue. I think in the research it was more basic insight within the exercise field that was the focus. But then of course with various diseases and the recognition that exercise was beneficial for various conditions, then it became more and more sort of into the medical medicine area, treatment, prevention, and so on. And that was where the research money was also more easily available. And I think that's why the field have moved in that direction, also because the money is there. Yeah.
So why don't we talk a bit about, so we're going to be focusing on glucose uptake in the muscle. So why don't we talk about, you've got the regulation of glucose uptake during exercise, you've got regulation with insulin, and then exercise, and then the insulin action. Why don't you just separate that out a little bit here for us?
Yeah, it's a little special, right? Because exercise is a sort of a catabolic stimulus while you're doing the exercise, whereas the insulin is an anabolic stimulus. So already there you can see a conflict if you sort of have at least both of these situations at the same time. But physiology, of course, try to avoid this during exercise by shutting down insulin release and insulin concentrations in the blood. So the issue is that, of course, there are some people that are treated with insulin, and they do not have this optional to decrease insulin within their body during exercise, and that's for instance type 1 diabetic, and for them this situation is problematic. So I think that's sort of that's one of the issue. The other issue is, of course, when you go into exercise recovery, where you would likely have an anabolic conditions. There you could see that whatever exercise have initiated, it would be beneficial to promote insulin action and the anabolic effect of insulin action. So you could see it as a problem if those both stimuli were present together, whereas in the recovery period, it may be beneficial that it have some influence on each other.
That's true. So maybe if I just summarize that a little bit. So you're talking about that during exercise, insulin actually goes down because you're having the contraction pathway for the glucose uptake mainly, and you don't want the insulin to be, taking glucose uptake all over the place, or even like, as you said, having an anabolic effect. But then after the exercise, especially when you have a meal, your insulin goes up, and then that's going to help with what your glycogen resynthesis, etc.
And it would be you could think of it as a beneficial thing if those muscles that have performed exercise were also those that benefited the most of the carbohydrate or glucose that were available. So in such a recovery period. So that's the way I would like to see it as a physiological phenomenon.
Exactly.
So, but the issue, when insulin was discovered in the 1920s, it was a wonder drug, of course, but it soon turned out that it was a major problem if you were physically active at the same time, and people went into hypoglycemia. And the reason is that, and there are several reasons, but one of them is, of course, that exercise itself activates glucose uptake, glucose disposal in the exercising muscle, and so does insulin. So suddenly you have two stimuli on board that increase peripheral glucose uptake. Also, normally during exercise, we increase hepatic glucose production to maintain glucose levels. But if you have insulin on board, this production is less efficient, and you actually inhibit hepatic glucose production when you have insulin on board. And therefore, there will be a balance with less glucose production, increased peripheral glucose disposal, and people will experience, or at least have a high risk of experience, hypoglycemia. And already back in that time, that became a major issue, and people or doctors did not encourage people to become physically active if they were treated with insulin. And that went on for many, many years because it was not understood, and it was because it was hard to manage the insulin dose and so on if people had a very physical active lifestyle. And it's even now 100 years later, it's even a problem to manage this for type 1 diabetic subjects.
Yeah, so we had Michael Riddell on the podcast, one of the first ones, and we talked a lot about type 1 diabetes and exercise. So that's one. If people want to hear more about that, they could go to that.
Sure.
So how about we think about maybe we think about the normal physiology, and then maybe talk about even the people with insulin resistance, which is a sort of more the type 2 diabetic person,
Yeah. So part of the research that was performed, turned out to show that that if you were physically active, you improve insulin action in the peripheral, and that was both if you did exercise training, but it was also following a single exercise part. And as you know, in obesity and type 2 diabetes, peripheral insulin resistance, lack of insulin action, is a major problem, or is part of the problem. And of course, if you could increase the action of insulin in the skeletal muscle, then you could think of it as beneficial for the disease, or for the prevention of the disease, and so on. So that's why it has been coming from this you could say, very small area of type 1 diabetes, insulin on board, and so on, to a much broader application of exercise in health benefit.
Yeah. So if we think about people that are insulin resistant, so just to explain to people, so someone that's insulin resistant has low insulin sensitivity. It's just a it's a different way. You can say someone's insulin sensitive. That means they have low insulin resistance. Someone who's insulin resistant has not much insulin sensitivity. But anyway, it's the same thing.
Right.
Now, if we think about that, this is why exercise is so important. I mean, you've done a lot of this work. You've shown that people with type 2 diabetes have normal glucose uptake during exercise, and they also tend to have a normal increase in insulin sensitivity after exercise. So that, they're obviously lower, but they tend to have a normal response.
Yes.
I want to flesh that out a little bit.
But that's fully correct. So Jorgen and co-workers in the 70s showed that insulin resistant or even type 2 diabetic subjects, they had a normal glucose uptake during exercise. You could have imagined from a mechanistic point of view that such two stimuli like exercise and insulin, they both increase glucose uptake in muscle by recruiting transporters of glucose to the cell surface membrane. And they actually both recruit the same transporter to the membrane. And therefore, this whole discussion of if one is not working as good as an insulin resistant, insulin is not working as good, why could it be that exercise did also not work as well? And that turns out not to be the case. And in fact, Jorgen and co-workers showed that actually glucose uptake during exercise in type 2 diabetic could actually be higher versus in controls. And one of the reasons is that they, in their study, they had, when they entered the study, they had high glucose level in the blood, and that probably have driven some glucose in. But it's definitely not decreased, at least in the studies that are available. And you guys have also performed some of these studies. So it seems to work very well. The resistance is specified for the insulin and not for the exercise part. Yeah. And that's, you can think about that as a good thing, of course. Absolutely. And then we have this phenomenon where, that you improve your insulin action also when sort of in the period after exercise or in recovery period. And that seems also to work well if you are insulin resistant. So if you are type 2 diabetic subject, you have a lower glucose disposal. And if you do exercise training, you can improve this phenomenon. You can increase your sensitivity. But the studies that have been performed is not that it's normalized. It's not a rescue within 10 weeks or 12 weeks or how long time we now do the studies. But it might very well be that if you perform lifelong training, it could be a rescue mission. That's so many other things. There's the beta cells and so on that also have. Yeah,
so the idea is, as you said, in general, it appears that even though they get sort of a normal increase in insulin sensitivity with exercise, it's still lower than the people that don't have diabetes. So what you end up saying is it's obviously better to prevent the diabetes than try and rectify it.
Yes. So you could say the diagnosis of diabetes, also depend on a defect in the beta cells. So you
which is the pancreas, just to clarify.
And you could say that if you lose the cells that produce insulin, and that's sort of where the time where the diagnosis is sort of defined, and you get hyperglycemia, and so in order to be a rescue mission, you, of course, we think we also need to improve not only the peripheral, but also the pancreas and the insulin secreting cells. And in some studies, you can see that may be what is happening. But I think we really need some very long-term studies to really prove this.
Yeah.
Yeah.
All right. See, so you've done a lot of studies looking at the mechanisms of how exercise increases insulin action at the muscle. Without going too complicated, do you want to talk about that a little bit? So especially I think around glycogen and the nice work you've done on that.
Yeah, so I think people have to, so I think we need to think about exercise as more than just one thing. So if you perform exercise training, you of course have to repeatedly go out and do some exercise bouts. And it's very efficient if you do exercise training. It works to improve peripheral insulin sensitivity within a few weeks, and in some studies within 1 week if you do very intense exercise. We can talk about that later, but every time, so that is one mechanism that sort of something is happening due to this training that improves peripheral insulin sensitivity. But one other thing is that every time you perform your exercise bout, then in the recovery period from every single bout, you improve insulin action as well. And it seems like that the mechanisms for these acute effect, you could say, on insulin sensitivity, they are different from those that you could call are part of the exercise training response. And to be a little academic or technical, you could say that exercise training seems to improve your insulin action or potency of so the dose response curve shift upwards, whereas with acute exercise bout you shift the sensitivity, so you shift the curve, the dose response curve to the left, the muscle becomes insulin sensitive. And for me, that's indicate that it's different mechanisms that are involved. So you have to imagine that if you perform exercise training, you improve your sensitivity over time. But every time you go out for a run or whatever you're doing, you improve your sensitivity. And in fact, the acute effect, it of course goes up and we're down. But if you sort of look at sort of the maximal obtainable sensitivity 4, 5, 6 hours after, it's much higher than what you gain with exercise training per se. So it's a complicated phenomenon. So you have these two things to think about. So with exercise training, I think that the literature will tell us that you get improved muscle mass, you get improved perfusion of the muscle that have been trained, you improve the quality of the muscle fibers, and with quality I mean the proteins content. Specific proteins are upregulated, and this could, for instance, be within the glucose field. It could be the glucose transporters, it could be hexokinase that are processing the glucose that is taken up, and it could be glycogen synthase that are regulating the glycogen synthesis within the muscle.
Okay.
So it's
so
more,
can I just before we lose you, so if you think about glucose getting taken up, so you mentioned hexokinase, so that basically, it's called phosphorylation. It adds a phosphate to the glucose, it goes into the cell, and then it's pretty much trapped in there. So you're saying we know hexokinase increases, the enzyme does that. And also you said the glucose transporters that actually go to the membrane and bring it in, they increase with training as well, yeah? Right. So yeah, just making sure people are clear on that.
And when you have glucose within the cell, what should you do about it? You can either burn it for energy, or you can store it for later. And normally when we have glucose available in the body, you also have insulin, and insulin is a driver of storing glucose within the cells. So 80% or so, 70–80% of the glucose that is taken up is going into glycogen to be stored. Glucose is stored as the molecule glycogen.
Yeah. And you're also saying that, so just again, you're saying that each acute bout, so each bout of exercise, you get an increase in insulin sensitivity, then it goes down, and you do another bout and it goes up and it goes down. And then you're saying training sort of adds on to that. So if we just think about that sort of time course there, is it right that each bout increases insulin sensitivity for like 24 to 48 hours. Is that
yeah.
Depending
so
on the
there
balance.
have been a few studies, there has been many studies, but a few studies where you have gone beyond 24 hours, and we have at least one in Copenhagen performed 48 hours after exercise, an acute exercise bout, where enhanced sensitivity was recorded. So it's a long-lasting phenomenon. In humans. In the rodents, which we use as model system, right? This is much shorter. It's a matter of hours, and then this effect is lost. But in humans, maybe for 48 hours. So the exercise bout you this Saturday morning, Glenn, you know, it's still remembered in your body. So you are sitting there with enhanced insulin sensitivity because you did something Saturday morning, right? Yeah.
Which is great. It kind of fits with the, I know we talk nowadays more about just trying to accumulate exercise every day or whatever, or physical activity, but you know, we used to say exercise every 3 or 4 times a week. So that would fit quite well with, you know, topping it up every couple of days. Or,
Yes. But you know, another issue that we need to remember. So when you perform exercise training, you gain a lot of benefit, including increased insulin sensitivity. But if you lay down in bed just for 3, 4, 5 days, you have lost everything. So it can take you weeks to gain, and it takes you just a few days to lose it all. And that's why the regularity of training is so important. No matter how much you do, just you're doing something. I think it's that's very important. And you know, studies where we have looked at what happens if people go to bed rest, and also Fleming-Dela in Copenhagen have looked at this. You know, just 3, 4, 5 days, then you have lost this beneficial effect.
Yes.
Yeah.
So what about people are interested in, you know, they always ask, what kind of exercise do I need to do to increase my insulin sensitivity? So, you know, how long, how often? Well, we talked about how often a little bit. How hard? What kind of exercise? You know, strength, endurance. You know, can you help us out with that a little bit?
Yeah. So in general, if you take sort of the big overview, I would say that the exercise that you actually perform is the exercise that are good for you. So all the thinking about what you should have done, etc. So I don't think that it matters whether you go for a walk or whether you run, as long as you just do some exercise, you will benefit of that exercise. So the body adapts to whatever challenge you put forward. So. But of course, with insulin sensitivity specifically, you could say, what do we actually know? Well, we have had with exercise training, we have had some studies where we had sort of asked the question, how low can you go? How little is sufficient to increase sensitivity? And together with Fleming-Dela, we did a study where subjects performed a quite low amount of exercise. It was sort of weight resistant type of exercise, but it was only 3 times a week, and it was only for 15 minutes or so, and the intensity was quite low. But that was sufficient to increase insulin sensitivity. So I don't think it takes much. Can I improve my sensitivity if I ask somebody to move my leg passively? I don't know. So you probably need to use your. I would guess you need to use your muscle.
What about that idea, though, that at one stage there was this idea in my mind, anyway, that you sort of break down the glycogen during the exercise, and then you resynthesize that glycogen, you know, basically back to the same point. I know you've done some studies with supercompensation, but so what about if you don't actually break down that much glycogen? You'll still increase your insulin sensitivity?
Yeah, that's a good question, and I don't think we have the answer, Glenn. So for a long time, we thought that the more glycogen you used, the higher your insulin sensitivity afterward became afterwards. But we have just performed some meta-analysis on 13 studies we have done, and we don't see this correlation. So we were quite surprised, because in animal studies there is this sort of association that the more you use, the higher sensitivity. But in the human studies we have performed so far, we don't see this. So you gain, no matter how little glycogen the subject have used, you still gain insulin sensitivity, and the increase seems to be quite the same, whether you use a lot of glycogen or little glycogen. But what we do not know is whether the use of glycogen determines for how long time you are in an insulin sensitized state. And that is something that I would like to ask either my colleague or the young people out there to start to investigate, because that may be the relationship between glycogen and insulin sensitivity.
Yeah, because that is surprising, isn't it? Because, you said earlier that the exercise, when you exercise, you use glycogen, you turn on glycogen synthase, which is an enzyme that's involved with synthesizing glycogen, as it sounds. And you would think that and then as the glycogen gets resynthesized, the glycogen synthase activity goes sort of back to normal. So you'd expect that relationship to be, sort of a dose response, wouldn't you?
Yes, yes. And that has, we have been arguing that for 2 or 3 decades. But those data that will come out here in November in diabetes, they will tell us at least that it's more complex than that, and that we should think about this phenomenon in more details. And so but it may not be. So what is important, is it the degree of sensitivity, or is it more how long time you have this improved sensitivity? So maybe I think, sort of from a health perspective, the latter is probably more important, and you get a huge increase in sensitivity. So So
was that after acute exercise? So with the meta-analysis,
this was after acute exercise.
Okay. So like you say, it might be similar, and then it doesn't last as long. Yeah, I get that.
Yeah. So we did this study where we compared the gain of sensitivity in the untrained state. So you do an acute exercise bout, how much do you gain? And then we looked in the same subject again after a period of training, where they have increased insulin sensitivity, a lot of positive effects of the training. And then we did the same exercise bout of the same relative intensity, and the gain in the trained state was less than in the untrained state. So there are some kind of interaction between the training and the sort of the acute bout of exercise. And from an academic point of view, you could start to argue, is it so that skeletal muscle can only obtain a certain insulin sensitivity? Is there an upper limit to how sensitive the muscle can be? That's an academic.
That's interesting as well, because when you train, you get higher muscle glycogen. So I was thinking they'd have to have more resynthesis to get the glycogen back up again. But
yes,
no.
Yes. And there are details that we don't know, Glenn. So in our study, they used the same amount of glycogen. But if you had, for instance, used more glycogen, had put up a training conditions or an exercise condition where you used more glycogen, maybe the outcome had been different. So what I'm saying is that we need more research on this.
Yeah, because I suppose, let me think, for a trained person to use the same amount of glycogen as an untrained person, they don't have to exercise as hard, because, the trained person, they spare their glycogen. So I guess, you know what I mean? Yeah. Yeah,
exactly. And you could also say, well, they after such a training bout, they still have high glycogen, and they have a lot of glycogen, so they are not in the same type of problems, perhaps, like in the untrained state. I don't know. It's
where things get complicated when you try and compare. Because I actually thought about this earlier, I didn't bring it up. But when we talk about people with type 2 diabetes having normal glucose uptake during exercise and normal increases in insulin sensitivity, the other thing I've thought about sometimes is we tend to use not your garden variety person with type 2 diabetes. We tend to use people that aren't on medications, You know, they might be just diet controlled. So then sometimes I wonder, can we actually say these things, you know, because what if you got people off the street that were on all sorts of meds and had high blood pressure and things? Are we confident they would have normal glucose uptake during exercise and normal insulin?
Yeah, but that's a good question. As a basic scientist, we try to control the most of our studies, and therefore we recruit very stringent, and so we have less variation and so on. So I don't know.
Yeah.
So, and that includes actually to take away medication from type 2 diabetic subjects, and in order we don't have this drug intervention interaction, and that's so
Exactly.
you ask a good question, and I don't.
It's a tough one. I think if we had the funding, the idea would be to do your person with type 2 diabetes on their meds, and you'd have some that have high blood pressure and some don't, and whatever, and you'd see what their glucose uptake was, I guess.
Yes.
All right. So, you know what, sometimes people think when you're doing research, everything works. You know, you've got 250, 300 papers or something. So it must be everything you've ever done worked. Is that right? Or why don't you give us a bit of an idea for maybe some budding scientists out there about real life? You know, what do things always work for you?
No. So I think success in science. Well, actually, I was teaching on a PhD course in Canada that last week, and there I took a quote from Winston Churchill, and he talked about success, and he says success is going from failure to failure without losing enthusiasm. And I think it's the same with science, because you can have all these great ideas, and often they prove not to be correct. And therefore you need to keep on pushing, put up new hypotheses, and then eventually you may create a picture where you sort of can say, well, it's likely that things are working in this way or the other. And do and you can seek your success. You can dream about success sitting there and just dream about it, or you can work hard at it. And I think it also takes a lot of hard work, and it takes a lot of luck to have sort of success. But you also need to have good ideas in order to sort of progress. And but definitely not. No, I have done a lot of failures, and I have done a lot of mistakes. Stupid mistakes, and the good thing is that I'm open about it, and I learn every time I do also stupid mistakes.
Exactly.
And that's sort of so you should also remember, Glenn, that we as scientists, we can never prove anything, but we can make it likely that things are the way we describe it, but we can never prove anything.
Exactly.
And that's also why I'm a little concerned about the science and the way we report science now. I call it storytelling. I mean, we have a lot of storytelling in science. If you can take a lot of papers in, let's say, high-rank journals, and they all tell you that they have solved the problem. So, and we all that are in the area know that that is not correct. But I don't know who pushes for this, but this storytelling where you are showing the data that support your hypothesis rather than discussing those that did not has become a trend. And I think it's a big issue in science, and the way we have discussion. And as you know, in the end, it's a matter of interpretation of data. You can have one interpretation of my data, and I can have mine, but we should be able to discuss this, and that's where we progress, when we have a dialogue about different views on how to interpret data. But I don't think the journals, some of the journals, are not allowing us to have these open ends in the stories, and that's a pity. And I really think that people like me and you and others are part of the solution, but we are also responsible, and we should.
Yeah, I know what you mean. They kind of want this tidy story. I've always had this idea that you shouldn't be able to write the introduction to a paper, because that's the rationale. Why did you do it? So, you know, we know this, we don't know this, and therefore, boom, 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 we hypothesize, boom, boom, boom. And then no matter what you find, you shouldn't change that. That was still the original idea, right? You don't like go back and change it to make a nice story out of it.
Right. So I can tell you, so I was at this, I've been at several international PhD courses where these young bright talent, they have these posters or presenting. And so over the last year, very, very few have a hypothesis on their posters or in their presentations. They have an aim. They have an aim, and but they don't pose an hypothesis. So you don't know. You can stand there and say, well, is it good or bad that you show whatever? And they don't know, because they just had an aim to describe something. And so I think it was like 80% did not state an hypothesis. So in theory, you don't know whether you should sort of what kind of conclusion you should draw from your data, right?
Right.
So that, I think that's a big problem. Of course, with the of course you can do research where you are hypothesis creating or seeking new hypotheses with omics techniques and so on. But far the most of the research that we are doing today is still hypothesis driven. And I am a little afraid that the young people are sort of afraid of stating hypothesis, because then it may be that you have to reject your hypothesis, right? And that's maybe, I don't know how they look at it, but I think it's something we should think
I wonder if part of it is this fear of these so-called negative findings. I hate that term even. I think it's, if you've done the study right and you get a finding, that is the finding. It doesn't have to be in any either direction. You know, it is the finding. I wonder if people are scared to sort of, you know, oh, I got a negative finding, I'll never get this published or something.
Yes. Yeah, it's terrible. And why is that? That's because the bigger journals would like to have these stories where everything just fit.
Happy ending.
Happy ending. Exactly.
All right.
So yeah.
I wonder if we could just talk a bit more. Okay, so with this exercise and insulin sensitivity, can we talk a little bit about sort of are there sex differences with aging? Are there sort of things like that we can consider a little bit, or?
Yeah, so you know, science have been biased to the direction of the male sex, and it's something that are, you know, being discussed a lot now. Through actually, we do know a lot of 50% of the population and very little about the other half, and that's of course a challenge. And I think more focus have been also from the funding agencies have been forced us to think more about this, and I hopefully in the, you know, in the next generation that this will be handled. But also within my area, we have used healthy male subjects, or these male subjects, young male subjects. So we have been very biased in the selection. We have done a few studies with acute exercise, also in females, where it seems like they have the same sort of beneficial effect of a single bout of exercise. And we also have, there are also female training studies that suggest this training will improve sensitivity. So it is actually so that female muscle in female, they are more sensitive to insulin. If you try to sort of match male and females from a lot of parameters, females seems to have a better insulin sensitivity in the peripheral, and that may change after menopause due to the changes in the sex hormones and so on. But until that, it seems to.
That's interesting. How is that? Because, you know, we talk about muscle mass being important, and you tend to think females would have. Is that per? We're getting a bit nitty gritty here, but is it per kilogram of muscle, or? Yes.
So that would be per kilogram of muscle, and it will also. So you know, the nature is so that the amount of fat percent-wise is larger in the female. So also, despite of a higher fat percentage, that this is observed. So that's something. So that's good. And but we don't, you know, the details, the mechanisms. I don't have any good suggestion for that. We should look for that.
And then just thinking about
with aging, what happens with aging? Well, you have loss of lean body mass, and that will probably tend to decrease insulin sensitivity for your whole body. You also tend to be less active, and if you are less active, you have less of these positive effect of being physical active, and that's that gives you a lesser insulin sensitivity.
Yeah, so that's what I was wondering. I wonder how much of it is the lower physical activity. It seems like a lot of things, when you look at the reductions with aging, a lot of it is the reduction in physical activity, up to a certain point. I think mid-70s things really sort of drop off. But
right. So and actually just dropping back to type 2 diabetics, I also think that's a major problem in obesity and type 2 diabetes. They will, in average, be less physical active than leaner subjects, and that is a major part of this insulin resistant as well. And I think also if you want to study the literature on this, you really need to be careful to see how well matched the controls and the obese or type 2 diabetic has been on a parameter slots like physical activity, fitness level, and maybe also lifelong, or the sort of the history of leisure time activity and so on. I think you need to be very well controlled to be able to conclude something on these studies.
Yeah, I mean, that's the feeling I've had. I remember even when I was looking at some cardiac stuff at one stage, and I know we had Ben Levine on one stage. It seems like quite a lot of what you look at with aging is just being less active. Yeah. Just thinking for people at home wondering, I wonder if I'm insulin resistant, or if I've, how do you go about testing that?
Yeah, so that will of course be somewhat difficult for people at home to measure directly. So we have methods in the lab that at least we call the gold standard, and the way we can do this with insulin clamps and so on, and that will give a good measure of how good your peripheral tissues like muscle and fat are to take up like glucose. Then of course you can do a lot of indirect measures, and also at home you can start to do measuring blood glucose, for instance. But people of course have to remember that blood glucose is that will be elevated if you are insulin resistant, more likely, but that's not only dependent on the peripheral, it's also dependent on the hepatic glucose production. It's also depending on the well-being of the pancreas and so on. So that's it, definitely not so easy to measure. So I don't have the good answer for that, how people can judge this. Then you could say, well, bio-tracking and the continuous glucose monitoring could give you some more dynamic hints to how well your blood glucose are responding to insulin or during a meal and so on. And there could probably have some indication of whether you are in a state where you are responding less to a certain kind of intervention or a meal or something, whereas in another period in life you could compare that and see, okay, now we are regulating glucose homeostasis much better to a challenge of sort of the same meal. So in that way, if you're sort of into this and you want to be a bio-tracker and so on, and have some interest and some skills to look at such data, you might be able to get some hints towards this. But
it wasn't long ago where people would just sort of go in and have a fasting glucose measurement. That's probably not overly useful, do you think, in terms of the dynamics we're talking about?
No, I don't think so. That will just give one measure in time, and it will depend on a lot of factors, so you cannot tease out which of these factors are important.
I guess even if you do, so some people may be aware of like a glucose tolerance test where you drink 75 grams of glucose and you follow the glucose over the next couple of hours.
Right.
But even that, unless you measure the insulin, you don't really know what's going on, right? With that.
No.
Yeah.
So, but you can use, as I try to say, you could use that for you know, before after training, and then compare yourself if you were sort of if you were interested in that. So then it might give some ideas, but not where these these changes take place. We have no idea. So yeah.
All right, great. Tell me, what are you working on at the moment? What are you excited about?
Yeah, so we are actually doing some very exciting stuff at the moment, and we are doing this in humans. And some of the things that led us on these thoughts was the variation that we are seeing when we are studying human subjects. Even though that we try to match them for a lot of parameters, it could be healthy, male, young, no disease, no family disease, etc. Some of the parameters that we are measuring has a huge variation between these subjects. And this, of course, also, in my case, this included insulin sensitivity within skeletal muscle. It varies like 10 or 15-fold within subjects that are healthy. 10 to 15-fold different? Yes. And that's very surprising. And also sort of the effect of a single bout of exercise varies a lot. Some increase sensitivity a little bit, others have a huge effect. And this is, so you could say if you do research in the traditional way, such variation is actually quite annoying because it creates variation, there is variation, and it's hard to find differences with interventions or between people and so on. And then, as you know, I'm a molecular physiologist, and I've tried to understand the mechanisms behind these phenomena as insulin sensitivity, and we have looked within cells to see what could explain that exercise improve insulin action, and we have some ideas, but it has not been so well. We have to some degree some success at the moment, but you could say that there are thousands of proteins within the cells, and these are modified by many different ways, both localization and activity and so on. And now we have methods to measure a lot of these proteins and these modifications in the so-called omics type of analysis. And it turns out that when we do these omics type of analysis, this phosphorylation of proteins, we see the same individual variation. So your phosphorylation pattern in your muscle will be different from mine. And we had some ideas that, okay, if these proteins are important, or these regulatory mechanisms, if they are important, they need to be associated to sort of the phenotype that you are interested in, glucose uptake during exercise, during insulin, and so on. So we have come up with sort of a new methodological ideas. We call it personalized phosphoproteomics, and we can sort of give special profiles to different people, and we can use this to associate to the endpoint of interest. And in doing so, we have actually used human studies to identify new mechanisms, new targets that could be involved. Of course, we need to prove this in subsequent studies, but that is extremely exciting because we can now use this kind of searching for mechanisms, and we can do that within humans. And that's I think that's where the future could one of the future paths that I will take is to take advantage of this. It is really complicated, right? So to clarify for people, you take a muscle biopsy and then you look at these, what you said, phosphoproteins. So it's the proteins that have had a phosphate group added to them. And there's like thousands of, so during exercise, during insulin, you have thousands of different proteins that are phosphorylated, yeah? Yes. So we, and also with insulin. So with insulin, we find that 2,000 phosphosides is changed just due to the stimulation with insulin, and that's so many that you can sit there and look at the data and you can get quite frustrated because how can we handle this? And then we said, well, among these 2,000 changes, some of them may if we believe in our hypothesis, some of them will explain our phenotype of interest, and that could in this case be glucose uptake. And we have sort of reduced this massive amount of data to a few, maybe 100 phosphorylation sites that potential could play a role, and therefore we have narrowed down from 2,000 phosphosites that was changed now down to 100 phosphorylation sites that are associated with our phenotype, and we can use that to guide us to be even more specific, to see, okay, then we will investigate this protein and this protein and so on. Mm-hmm. But the interesting thing is that this happens in human beings and not in animal models. So it's a huge step forward.
And are you able to, you said there's 10-fold differences sometimes between people. Is that able to be matched up with your phosphosites, or is that a bit hard to?
Yeah, so the paper we published in Nature Biotechnology, we did not bring in. We used some. We try to calculate or try to develop a model and an algorithm so that we could we predict from the phosphoprotein from what we measured in the muscle, could we then predict how well insulin works in your body. And the criticism was that it was on a very small data set, but within this data set and with the modeling that we did, we could actually predict it quite well. So it has huge potential, and I think in the future you will see that that you can use this in other diseases. If you can apply it in cancer, for instance, or other diseases, you could also think, that's what we are going to do now, to do this in bigger cohorts, and also to see if we can figure out what kind of insulin resistance do we see in type 2 diabetes. Are they all the same at the muscular level, or are they different? So that's really what I'm excited about, because
that's pretty cool.
Yeah.
Just one thing I was just thinking we hadn't really talked about, because you were talking about with disease, is can we just flesh out a little bit the importance of this insulin resistance, like for disease? So we've talked about people with type 2 diabetes being insulin resistant, but this metabolic disorder, it seems to be quite central to a lot of disease. Yeah.
Yes. So cardiovascular diseases is associated with insulin resistance, for instance. And so, but it's hard to say, what is the egg and what is the chicken. What do you say? Chicken. Yeah. Chicken. So, but a lot of these kind of syndromes are associated with insulin resistance, but they are also associated with obesity. So which one is
even cancer, important? Maybe?
Yeah. And even cancer, of course, metabolic. Also, yeah.
So when we're talking about exercise, increasing insulin sensitivity, we're talking about, huge effects on the body potentially.
Yes, and various research groups are very keen in figuring out how this works and how does exercising muscle communicate. I can't say that. How does these exercising muscle talk to other organs, right?
Crosstalk, yes.
Yeah, so the crosstalk, and try to figure out how can, even cognitive capacity seems to be increased with exercise training and so on. So maybe it also have a role in maybe not preventing dementia, but maybe postpone developing of dementia and stuff like that.
Exactly. Okay, this has been great. I think we're starting to sort of get close to finishing up. We've talked about a lot of things, but on Twitter, on the Inside Exercise Twitter, I ask people, I say, Jørgen Wojtaszewski is coming on. Do you have any questions? And a guy we both know who's actually done research in Copenhagen, Dr. Adam Rose from Monash, asked an interesting question. He said, you and others have done excellent work detailing activation and functions of AMP kinase, AMPK, complexes in skeletal muscle with exercise. In your opinion, is it safe to drug AMPK in absence of high energy burning? So, is AMPK a drug target type of thing? What do you think?
Right. Yeah. So that's a very good question from Adam, of course, and it's something that a lot of people have been thinking about. Of course. So you could say, Adam, that that if we treat animals, we can enhance. If we treat animals with AMP kinase agonists, then we can show in preclinical studies that glucose homeostasis is improved in animal on a high-fat diet. There are several studies on this. So this would suggest that it has some potential, of course. And so in a sense, that's good. And of course, AMP kinase in itself will not increase energy turnover. And therefore, activation of AMP kinase is not a cure for obesity or energy surplus. So in that sense, it's not a cure like that. But you could say in type 2 diabetes, it is caused by an imbalance between how much glucose is taken up in the peripheral and how much glucose is actually produced by the liver. And you could say that if you could improve the peripheral insulin sensitivity by dropping or drop AMP kinase, or use it as a target, then at least that part of the cause of imbalance is taken out of the equation, and that will probably normalize the sort of the imbalance. But of course, it will not take away the original cause of the peripheral insulin resistance. So it may be a treatment, but it's not a cure. Perhaps in the long run, we don't know, but immediately it's not a cure for type 2 diabetes.
Yeah. Yeah. And also, I guess, even if it is, so it's stimulating glucose uptake. Yeah. So it could drop the glucose a bit, but obviously exercise does a whole bunch more than that. So
Yes.
As you've alluded to, it's funny because if you email you when you're on holidays, it says you're chasing the exercise pill. It's like a bit of a joke. But how far off do you think we are with that? And realistically,
we are far away. But someday it may come. We are of course optimistic, Glenn, in the sense. So it's a part of a joke, and but you could take the parts that you sort of, if you find the mechanisms for some of the good things that exercise is doing, you may be able to find also drug targets in those pathways or those mechanisms that you sort of can hit with upcoming new drugs. And AMP kinase could be such one, right?
But us exercise physiologists obviously think just go out there and exercise. Yeah, you're going to get these things. And
yes, that's the way to do it. And as I said before, all exercise that you perform is the exercise that's good for you, right? And
exactly.
The exercise that you don't do.
But it is true. Good. It is true, I guess, that I tend to think just go out there and exercise, and we know that's easier said than done. But I had Paige Geiger on here talking about heat shock proteins and things, and she made the point, which is fair enough, that not everyone can exercise for various reasons. It could be injuries or just general health. So, if you can take a drug, or as she would suggest, jump in a hot tub to activate all these heat shock proteins, that's one way of getting it done. All right. So yeah, thanks for that. So I guess before we go, what about just take-home messages for people? So just sort of summing everything up, what's the take-home message for people at home?
Yeah, so you could say within my field, exercise seems to have a lot of beneficial effects and health beneficial effects, and sort of as I just said. So go out and do exercise. It will definitely be good for you. And I don't really think it's a matter of what type of exercise that you are doing. Of course, you will have different outcomes, whether you do strength training or endurance training, but in a matter of sort of health, I think all the exercise that you do is sort of good for you. And in the long run, we are chasing sort of the mechanisms behind these beneficial effects, and we are not there yet. We have some clues to some mechanisms, but there's a long way, unfortunately, to go yet. But once we have the mechanisms, we may be able to guide people better towards the right exercise for a given population, and so on. So
great.
I think that's the major change, and exercise is good because you increase the whole body insulin sensitivity, and that is good because insulin resistance is a bad thing, and it seems to associate with a lot of diseases that we try to avoid. So
yes,
go out and hit the exercise.
And also if people, you know, miss a day or whatever, don't feel bad about it, because each bout of exercise increases your insulin sensitivity. So, you know, sometimes people go, oh, I started an exercise training program, I've missed a couple of days, you know, but it's like, hang on, every bout of exercise increases your insulin sensitivity. So just go out there and do it.
Yes.
Yeah? Exactly. Okay. All right. Well, thank you very much, Jorgen. It's been great. And
thank you, Glenn.
I'll see you when I'm back in Copenhagen next year, hopefully. Okay.
Yes, hopefully. See you around.
Mate. Okay.
Yeah.
Bye-bye.
Bye.